Assalamualaikum warahmatullahi wabarakatuh. My name is Nur Shahada binti Abdul Malik from AS2446. My title is Analysis of Caffeine on Case-Based Gene Regulation using Human Gastric Carcinoma FSG671. This is the overview for the slide. Caffeine is natural alkaloid compound that isolated from coffee beans and chemically known as the 1,3,7-trimethylxanthine. Caffeine can be naturally found in various of plants such as tea leaves, cocoa beans, kola nuts, guarana berries, and many more. It is functioned as the stimulant to increase the activity of central nervous system. Caffeine properties is an anti-cancer that can activate the pathway of caspase that lead to the apoptosis. Next, caspase-3 is a family of protein from cysteine protease family. Caspase-3 is an execution of caspase which is play a roles as programmed cell death and also known as apoptosis. Caspase-3 can initiate the death receptor stimulations for cancer cells. Gastric cancer is a disease that is from the uncontrolled and rapid growth of abnormal cells that can occur from the numerous interactions, for examples from environmentals, host factor, and genetic factor. The problem statement of the study is caffeine is able to induce apoptosis by activations of caspase 9-03. Previous study has been reported that treated cancer cells with various concentrations of caffeine will inhibit the cell growth and reduce the viability which caffeine promote gastric cancer cells apoptosis. The objective of the study is first to determine the caspase-3 gene regulations in treated and non-treated HGC27 cancer cell line in vitro with various concentrations of caffeine. Second, to verify the implications of caspase-3 gene regulation in HGC27 by DNA sequencing analysis. The methodology begins with the first is the primer design. Second is the preparations of the complete culture media with the revivals of the HGC27 cancer cells. Third, the cells feedings and the cells growth. Next is the treatment of the HGC27 cancer cells with caffeine for 24 hours and 48 hours. Then, RNA extraction followed by the determinations of RNA concentrations by using spectrophotometer. After that, the determinations of the RNA integrity by formaldehyde agarose electrophoresis and reverse transcriptions into cDNA. To answer the first objective, the amplifications of caspase-3 and beta-actin by PCRs and gels electrophoresis of PCR product have been conducted. After the objective 1 has been achieved, the gel extractions and the determinations of purification of the DNA procedure have been conducted. It is to obtain the objective 2 by DNA sequencing. Result and discussion. Based on the result obtained, the morphology of the cells that were not treated with caffeine or control treated with distilled water is look like a spindle in shape and polygonal shape before and after 24 hours and 48 hours. The shape is remain the same and unchanged. For the cells that's been treated with caffeine, the morphology of the cells change with the shapes look like elongated spindle shape and the cells adherence to each other. The elongate of the cell shapes can be seen more in 48 hours after the treatment with the caffeine. Next, based on the gel electrophoresis result by using beta-actin primer. From the figure, the DNA bands of approximately 168 base pair, which is the targeted size for beta-actin genes, was obtained. In the figure, it shows that for beta-actin primer, all samples have been treated and control untreated with caffeine have the same intensity. The intensity of the band is high. So, it gives us a positive expression. This is because beta-actin acts as a constitutive gene or a housekeeping gene that perpetually express the expressions. So, it indicates that the cancer cells are not undergoes apoptosis. Next, we move on for the caspase-3 primer result. From the figures, the DNA band appeared on the gels approximately 161 base pair, which is the targeted size for caspase-3 gene was obtained. From the result, we can see that the DNA band that have been treated with various concentrations of caffeine faded, and it has low intensity, compared to the DNA band that has been not treated with caffeine. The intensity of the untreated DNA with caffeine is high. So, we can see the difference between the band intensity. It is indicate that the cancer cells that have been treated with caffeine undergoes apoptosis. This means that the caffeine activate the pathway of the caspase that lead to the apoptosis. So, it shows that the objective one from this study has been achieved. To achieve objective 2, DNA sequencing has been sent, but the result couldn't be included in the slide due to the several problems because the files couldn't be opened. In conclusion, the objective ones of the study is achieved, as the result shows that the caffeine activated the caspase-3 genes, so it is indicate that the cancer cells undergoes apoptosis. Unfortunately, objective 2 couldn't be proven due to the DNA sequencing result file couldn't be opened or couldn't be read. For the recommendations of the study, firstly, we need to increase or use various concentrations of the caffeine, such as 3 millimolar, 4 millimolar, or 5 millimolar of the caffeine concentrations. Then, increase the time frame during treating with caffeine, for example, 72 hours or more. Here are some references that have been included in the slide. Thank you for listening.